Everyone, welcome to today's webinar, Best Practices for Decontaminating Biological Equipment and Spaces. My name is James. I run our webinar and events program here at Triumvirate Environmental, and I'll be your moderator for today's event. Before we get started, I'd like to give you a brief overview on what the webinar is going to entail as we run through a few housekeeping items. You may have noticed when you logged on that your microphone is muted. We ask that if you have a question or experience a technical issue, to please communicate that using the questions box, and I'll be able to answer those questions. Following today's presentation, there will be an open Q&A session. You can submit questions throughout the webinar, again using that same box, and we'll get to as many as we can. In the event we don't get to them all, we'll be sure to follow up with you in the next couple of days to make sure your question's answered. And finally, please keep in mind you'll receive a copy of this slide deck and the webinar recording in an email tomorrow, so keep an eye out for that. Our speaker today is Ryan Miller. Ryan is an engineering project manager at Triumvirate Environmental, where he has conducted numerous environmental engineering projects and remedial response actions. This includes tank removals and closure reports, laboratory and facility decontaminations, hazardous building materials identification, and sodium azide remediation, just to name a few. Mr. Miller has also carried out engineering consulting services, remedial system operation and maintenance, building material inspections, and emergency spill response work throughout his career. He has a BS in environmental engineering from the University of New Hampshire. And quickly, before I turn the microphone over to Ryan, we've had some questions about the services we offer, so just wanted to take a few seconds to give you an overview of that. Triumvirate operates primarily within 4 markets: life sciences, healthcare, education, and industrial across North America. Our service categories include waste management and disposal, EH&S consulting, field services, and on-site EH&S support. Our services range from hazardous waste disposal and regulatory compliance, biosafety services, to lab decontamination and chemical inventory management. So I'm not going to read the whole list. If you're interested in learning more, feel free to visit our website at www.triumvirate.com. And without further delay, I will turn things over to Ryan.
Thank you, James, and thank you everybody for attending. Just going off of what James just said, with regards to my career, I've definitely seen over 100 different laboratory scenarios and decontamination projects with respect to biology and biological equipment and spaces and the decontamination of those. So today I just want to get my experience out to all of you with regards to what you can do as far as programs and procedures to maybe prevent having contamination happen where my services would be needed. So really try to stop this on the front end, be aware of where contamination sources come from and how you can prevent them. So with the agenda, we have contamination sources. We'll talk about some common biological equipment and spaces, a couple case studies, my examples of what I've seen, and then we'll follow up with some Q&A. So let me turn this over to James again with a quick poll question. When was the last time you had to decontaminate a room or a piece of equipment?
Thanks for teeing that up, Ryan. Just launched that poll. We'll see the options there for you. Within the past 2 years, within the past year, within the past 6 months, within the past month, and never that I'm aware of. So Ryan, we'll share those results in a few seconds.
All right.
All right, let's get a few more in. Okay, looks good. I'm going to close that, Ryan, and we'll push that out. So looks like 30% within the past month, 27% within the past 6 months, and then third would be we never that I'm aware of, 20% of our audience.
All right, that's good to know. Let's move on to potential contamination sources of why we would have to do a decontamination of a room or a piece of equipment. So causes of contamination, You have laboratory personnel, poorly developed or poorly executed procedures, which could be lab coats, mislabeling, cross-contamination, maybe through pipette tips, cross-contamination of poor cell culture management, unclean or dirty work environment, or poorly designed spaces. So what that can do is it can lead to mold, it can lead to dust or debris buildup, or it could lead to cross-contamination of media. That can also lead to mycoplasma contamination. So mycoplasma is the smallest free-living organism. It's considered to be the simplest bacteria and lacks a cell wall. And that's important because common antibiotics work by keeping a bacterium from building a cell wall. So the lack of mycoplasma having that makes it immune to those antibiotics. So what is mycoplasma contamination? It's commonly found in cell cultures after a larger contamination is present. Something like mold can lead to this smaller free-living organism of mycoplasma. And once that is in a space, it functions as a parasite to meet energy and biosynthesis demands. And it's hard to detect because it's so small, and it multiplies very quickly within cells. So where can it come from? Well, since it's ubiquitous and it has a broad range of hosts, it can come from really anywhere. Humans, birds, reptiles, fish, insects, and even plants have the ability to have mycoplasma contamination. So it was first reported in cell culture in 1956, but it continues to challenge scientists just because of insufficient protection and carelessness within lab spaces, tissue culture rooms, or any biological spaces. There have been no perceivable reduction of cell culture infection rates because of that. So even though the risk and consequences caused by mycoplasma infections have been known for decades, measures to prevent infection are established, many cell lines are still infected. And that again leads to carelessness or insufficient protection. So once mycoplasma is in a cell culture batch, it competes with host cells for nutrients. It can alter DNA, RNA, and protein synthesis. It diminishes amino acid and ATP levels and induces chromosomal alterations and modifies host cell plasma membrane antigens. All things that you don't want to happen in your cell cultures. So sources of mycoplasma in cell cultures, sometimes it's just infected cells sent from another lab. That's a common thing, unfortunately. Contaminated cell culture medium reagents, again, could be contaminated elsewhere and brought into the lab. And it can also be brought in by laboratory personnel who's infected with the mycoplasma. Unfortunately, once it's in, contamination spreads rapidly to other cell lines, and that will lead to the need for disinfection. So despite resistances, the addition of suitable antibiotics is best based on current and past research. Things like quinolones inhibit the enzyme, that DNA gyrase that is necessary for the replication of the bacteria. So that form of antibiotic is suitable. So if that cell line can be saved from the antibiotic, then you don't have to throw it away, which is a benefit. But if it can't be saved, the next option is to just discard it and start over. But if you do have a cell line that is contaminated and you get it either thrown away or improved by antibiotic, then you're going to have to attack the surfaces that came in contact with that cell culture line. So how do you do that? Surfaces like small tools or things that are able to be autoclaved, compatible materials, that's fully efficient. Other materials can receive ethanol rinse, 70% isopropanol is fully efficient, or ethanol, complete inactivation after 5 minutes of exposure. An alkaline cleaner works. Increased concentration increases efficiency, and they found inactivation at 8%. Chlorine dioxide and vaporized hydrogen peroxide are very efficient for enclosures in small rooms, and quaternary ammonium compounds have been found to have poor efficiency for mycoplasma disinfection. So mycoplasma is just one of the small things that can contaminate biological equipment and spaces. Another broad range is just gross contamination from dust, debris, or mold. So where will we find those things, and how can we prevent them? Incubators. Incubators are a big source for contamination because it is where cell cultures are allowed to incubate. So how do you prevent that from happening? Just from normal function, incubators' fans become loaded with debris. Buildup occurs on sensors and gaskets. Basic surface cleaning and gas disinfection does not remove that buildup, and that leads to contaminated media. So some incubators have self-clean functions, which is usually a high temperature, a UV light, which is useful in killing bacteria and may kill spores, but some bacteria and spores will need temperatures around 266 degrees Fahrenheit, and UV lights are not as effective at killing spores because the UV light must be in contact with that spore for an extended period of time to have any effect on the mold. So this is why it's important to remove gaskets, fans, and sensors and clean the hard to reach areas behind them. Most commonly, incubators are just opened, wiped down, and closed. There are many other components to that incubator where contamination lies and can only be cleaned by following that procedure. So the procedure is make and model specific. It usually spells it out in the user manual for that specific incubator. You should follow the method recommended by the manufacturer, which is most commonly replacing consumable parts such as filters, tubing, and gaskets. Porous materials, other porous materials in there, they're just hard to clean. So discarding heavily contaminated parts and replacing them with new clean products is sometimes preferred. Thermo Fisher even sells a PM kit, which includes filter casings, HEPA filters, gaskets, tubing, and rust inhibitor, and they sell that for around $400. So once everything's out, you just need to clean and replace parts and shelving, supports, plugs, grommets, ducts, humidity pans should be thoroughly cleaned, gaskets, filters, and fans. So how often should you be doing this? Well, this is a excerpt from a Thermo Fisher 3110, which is a common incubator in most scenarios. And it says that the CO2 tank levels should be checked daily. That should be obvious. But inspect door latch and hinges and the door seal. That should be done yearly. Check the water level in the humidity pan weekly. Verify and document all the levels in calibration yearly. And then here, complete decontamination procedure. And that involves wiping down the interior, shelves, side of ducts with a disinfectant, clean or change blower wheel and scroll, clean top duct, humidity pan, rinse everything well with sterile distilled water. See, cleaning the interior. So they have a more detailed procedure in the manual. And you see that it says it should be done between experiments. So that's something important to keep in mind too, is that you can have cross-contamination between experiments if you're not performing a complete decontamination of that incubator. If it doesn't have a clean environment to grow and there's some sort of other contamination, then you're not going to get the optimal results you're looking for. It says more frequent decontaminations may be required depending on use and environmental conditions. And that's where these other recommendations come in to change the HEPA and gas filters, if applicable, and installing air sample filters and things like that every 6 months. And again, doing checks on the hinges and gaskets and door seals every year. Another common biological piece of equipment that requires preventative maintenance and procedures is a biological safety cabinet, or BSC. So BSC contamination has internal parts and HEPA filters that may become loaded with debris, and basic cleaning does not remove this buildup. This can lead to contaminated media. So when is BSC decontamination needed? After large spills of chemicals or biological material, prior to any maintenance work, filter changes, and performance tests that requires access to any contaminated portion of the BSC. That would include the internal HEPA filter or any sort of flock filters before that HEPA. Decommissioning, disposal, or salvage. Obviously, want to eliminate any biological contamination before you're giving this away, disposal, anything like that. It needs to be cleaned so we're not introducing that bacteria into the environment. Or initiating a new work program or production run. That's again to just make sure your new run isn't going to be contaminated with something from the old. And relocation from one building to another. Again, preventing migration of biological materials throughout a building or common space. So the most common areas where contamination is found in a BSC during routine work would be the work tray. So typically at the end of a work shift or a class or a research event, wiping of that work tray is conducted probably most commonly with 70% ethanol or isopropanol. The problem here is that BSC is moving so much air during that process that it's actually able to pull things below that work tray out of visibility. You see the most common areas when I go to decontaminate a BSC by gaseous decontamination lies underneath that work tray, out towards the back where it's pulling in air to go through HEPA filtration. You get a lot of solids, dust, debris, pipette tips, things that you wouldn't even think of I've seen in the back of BSCs. That's the most common area where the contamination lies and can potentially contaminate the rest of your process. So the work tray is readily cleaned since it's stainless steel and accessible, but cleaning underneath is often overlooked, and the airflow from the BSC draws bulk materials underneath. So BSC decontamination, when it comes to gaseous decontamination, is instructed by the National Sanitation Foundation and the American Standards Institute, ANSI 49, Biological Safety Cabinet Decontamination Standard. And that procedure includes removing panels, front panels, bulk materials, and debris from underneath the work tray and other areas where things get loaded. Then the BSC is sealed, and in this case, a mini chlorine dioxide system is utilized to disinfect the cabinet. So Triumvirate uses this mini chlorine dioxide system. It utilizes chlorine dioxide gas for biological decontamination per the NSF and ANSI standards. And it requires a 90-minute contact time with an approximately 20 to 45-minute scrub cycle. It's fast and safe and decontaminates the biological safety cabinet and any internal HEPA filters within that time frame. Utilizing the unit's internal scrubber takes around 45 minutes to scrub the gas out of the unit, but we also have larger scrubber that can be attached externally that can do it in about 20. And this system can be piggybacked on multiple BSCs to perform multiple BSCs in a day. So what else is out there? The NSF has 3 methods described in their latest manual, and it's formalin, vapor, chlorine dioxide, and hydrogen peroxide. So this is just a small means of comparison here. And formalin vapor, nobody really uses anymore just because of its carcinogenic effect. Everybody's leaning towards chlorine dioxide, hydrogen peroxide today. The time per cabinet depends. VHP decontamination time is a variability attributed to whether or not the BSC is ducted. If it is ducted, VHP may be released into the HVAC if it's isolated to the roof, and obviously there's nobody working in the area. But if it's not ducted, it must remain aerated inside the cabinet and can take up to 10 hours. Whereas the chlorine dioxide, you have a setup time, a contact time of 90 minutes, and then approximately 20 to 45 minute scrub cycle. So that's about 3 to 4 hours. So that's just comparing the two. We can get deeper into comparisons here, just because it's a common question of what's the difference. Chlorine dioxide does have an odor, vaporized hydrogen peroxide does not. This table is based on using those systems to do large spaces like a BSL-3 lab. This was taken from Biological Research Academy, and the comparisons for each gas, they're just showing that, you know, indicators were utilized to demonstrate each method's ability to penetrate and distribute throughout the room and determine what size spaces they're effective to. And the study showed that chlorine dioxide gas penetrates better than VHP, and that's just because chlorine dioxide is a gas, whereas vaporized hydrogen peroxide is a vapor. So the vapor takes a lot of air movement to get into areas, whereas the gas is going to dissipate into everything. It'll even go through electrical outlets. You know, it's a gas, so it's going to get everything. So with that, I'm going to turn this over to James again for a quick poll question. James, you want to kick that off?
Yes, thanks, Ryan. And just like last time, we're going to give you about 20 seconds to answer this question. Which decontamination technologies do you typically use? And for this question, you can choose multiple options. And we'll give you a few more seconds and share the results.
Thanks, James.
All right, we'll get a few more in there. And looking good. So let's see what we've got here, Ryan. All right, 32% VHP and 45% other, and some use both, as you can see here.
Great.
Any surprises there?
Yeah, I want to, if you could, at the end of this, be so kind to email me what maybe that other is, just so I'm aware. All right, with that, I'm going to move into cold rooms. So what kind of contamination do we commonly see in cold rooms? Mold. Mold is often identified in cold rooms on ceiling grates, fans, light fixtures, metal racks, and it often occurs when the cold room is left off or allowed to warm up for an extended period of time. That can lead to gross mold contamination, but smaller level contamination can happen when materials are taken out of the cold room, allowed to warm up in the lab, and then brought back in. And that's because mold spores don't actually die when temperatures drop below 32 degrees. They're still viable, and they will grow once the temperatures warm up. So the air in cold rooms is recirculated. So if you have mold growth, spores are heavily distributed, leading to rapid growth. And mold needs a food source, so its common food sources include paper products and porous material. And once those become infected with mold, it's almost impossible to clean. A lot of times it just has to be disposed of. So mold needs moisture. And how does moisture enter a cold room? Well, condensation is the biggest culprit here. This can come from condensation on light fixtures or fans, leaky pipes, or water spills. In order to stop the problem, the moisture source must be eliminated. Many cold rooms have high humidity of 50% or greater due to condensation that forms, and that's just from opening the door. So since cold room is at lower temperature, the warm exterior air enters the cold room, and instantly the moisture in the air condenses, and it increases the humidity in that room, and then the door gets shut, and it happens every time. So every time the door is open, the more moisture enters the cold room. Some cold rooms can even reach 95 to 100% humidity, ideal for mold growth. Some cold rooms are equipped with humidity monitoring systems and dehumidifiers to prevent this, but it's very uncommon. So once you do have mold, what can be done? You have to eliminate moisture sources, remove all cardboard and porous materials, Use a biological disinfectant to clean all surfaces, and I recommend a HEPA vacuum or filter to be running in the space to capture airborne spores. Would also keep in mind cold materials should not be out of the cold room and allowed to warm up significantly while this happens, so transferred to another cold room or refrigerator. And consideration for isolation or keeping that contained, that cold room contained while this work happens to prevent mold spores from affecting the rest of your facility is definitely something to keep in mind. So once the cleaning has occurred and there's no visible or odor contamination, you can perform spore trap analysis or surface wipe samples. So confirmation can include these two analyses, and it's a typical method that uses direct optical investigation to determine the amount of spores in air or on a surface. In air would be spores per cubic meter. And with the analysis, you can also determine whether there's any active growth from a tape lift sample on a surface, and you can even identify the type of mold. So how do we prevent all that from happening in the first place? Routine maintenance, you got to be proactive. So set up a routine, eliminate moisture sources, porous materials, and be proactive with the materials being taken in and out. Consider any boxes or small kits be bagged in Ziploc bags to prevent mold from growing on the cardboard. And set up a routine. I recommend full decontamination biannually and a full decontamination between operational changes. Also a full decontamination if you do identify mold through a visual or a suspected odor. It's just best to do a full decontamination at that point as well. And that's going to lead to our next poll question for cold rooms. James.
Thanks, Ryan. Sharing that question right now. I think everyone, you're all pros by now. We'll give you about 15 seconds on this one and share the results. So far, looks like there's one clear winner. I'll give it a few more seconds. All right. Ryan, 58% selected never, and 17% every 6 to 12 months. I'm seeing only about 12% every 12 to 24 months.
Okay. I'm not too surprised there. And with that will lead into some case studies of why I'm not too surprised. Unfortunately, I've seen a lot of contamination in biological spaces and equipment, and that is mainly because there isn't any routines set up or preventative maintenance. So let's just go through. Here's some photos. We'll start off with the first one we started off. This is an incubator. This incubator was involved in a experiment that had contaminated a large batch of tissue cells. This was one of a dozen incubators that look similar to this, but when you open an incubator, you usually see all the shelving and the humidity pan and the brackets. This is what it looks like when all of that is removed and it hasn't been cleaned ever. They were identified as the source of contamination, and pulling off the removal ductwork, sensors, filters, water pan, there's clearly contamination in the incubator. The visible contamination obviously wasn't visible when the removal materials were in place. So, you know, it looked clean, but it wasn't. And it was actually the culprit of causing a lot of headaches and a lot of lost profit. So with that, routine maintenance without just doing the self-clean is definitely recommended. This is a close-up of the fan spindle. That's actually what's blowing into the incubator. It's blowing through a HEPA filter, but if you're not cleaning your HEPA filter, then this is just going straight through and blowing onto everything that you just put in there. So buildup of the fan housing underneath baffles, behind sensors, that just becomes loaded and gross. So full decontamination and consumable replacement will mitigate this issue. You know, what can lead to this? It's operational. So by only using the UV light or a high temperature cleaning, you're actually not removing any of this material, and the loading in the fan housing, that's pretty significant to contaminate whatever is being put into that incubator. So this is a after picture. As you can see, that's a new fan spindle, a new filter, new tubing, putting the shelves in. Looks a lot better, a lot cleaner, and a lot safer for whatever materials being put in that incubator. Just new consumable products, filters, tubing, and a surface decontamination using 10% bleach and 70% ethanol was able to solve that problem. So here is an example of a BSC with its work tray removed. As you can see, this came about when there was a complaint about contaminated media. There was thoughts of cross-contamination between animal rooms and tissue culture rooms. There was all sorts of discussion around it, and this is what I had found when I opened up one of the BSCs in that area. As you can see, over time, pipette tips, even unused, unopened pipette tips, had just been drawn in through the airflow of that BSC, had collected where that air goes up and in, and had just been sitting there and likely throwing around contamination and contaminating the media that was being used. So removing that showed just how bad the contamination was. We cleaned it, but after large spills, or if you're noticing that things are disappearing while you're working in a BSC, it's likely that they're being drawn underneath into that work tray. And it's a simple thing to just open up and look under and see just how bad contamination can be. This is another case study where a lab ordered a BSC decontamination, and what happened is the vendor came in, threw the stuff in on top of the work tray, turned it on, put the VHP through, let it do its thing, and then left. Came back, got their stuff, and left. A couple weeks later, they noticed contaminated media again, and I spoke to them, went out and looked, and none of the filters had been removed. The filter on the left is called a pre-filter paper catch. That it was obviously contaminated with just dust, debris, and whatever was being utilized in that biosafety cabinet. And then another one in that same area had significant loading materials underneath that work tray. So a gas decontamination can only go so far. If you leave bulk materials and debris, it leaves an area for contamination to grow back and then essentially contaminate the rest of the materials. So gas will only affect it so much. You need to have clean surfaces, visibly clean surfaces, in order for that gas to penetrate and to eliminate any smaller sources of contamination other than this bulk stuff. So using the mini chlorine dioxide system and wiping a thorough HEPA vacuuming of the interior of that BSC, we're able to get it clean. We're able to do about 5 to 6 cabinets in an 8-hour period. We've gone a little above that with our new scrubber. Can do about 8 in 8 hours. So it's definitely a beneficial service if you're looking to have some BSCs decontaminated. So moving on to cold rooms, here are some examples where cold room mold is found inside light fixtures, drop ceilings, on these chrome racks, metawire shelving, on light fixtures, on ceilings. So whenever that's identified, the material either has to be cleaned or in some cases when it's so bad, the materials have to be disposed of. So Triumvirate in this case conducted a decontamination in the cold room according to our SOP, and assessment activities were conducted for mold subsequent to decontamination activities. Our personnel uses the proper personal protective equipment, including full face air purifying respirators, Tyvek, nitrile gloves, booties. That's just to prevent any contamination to our personnel as well as contamination out of that cold room. We built the containment outside of the area to prevent any migration of mold spores. And all accessible surfaces, floors, walls, ceilings, shelves, benches, air handlers, shrouds, light fixtures, door seals were decontaminated. We removed the drop ceiling grates from the ceiling and cleaned them to the lights and the handler fans, and then conducted confirmation sampling to ensure the problem has been solved. So those are just a few examples of what happens or what can happen if routine preventative maintenance and cleaning is not performed. Kind of a worst case, but we see it a lot. And you know, again, I'm just trying to get in front of everybody to show that this can happen, and there are ways to prevent it from happening, and there's ways that we can also help. So with that, I'll turn it over to James for some Q&A. He's going to read me some questions.
Thanks, Ryan. We do have a few questions that have come in already, and we will open the floor. We've got plenty of time here, and we'll be able to address as many questions as we can. So we'll advance to the final slide, and I'll share some more information with you as you're typing your questions in. All right.
So,
do you want to cover these takeaways here?
Yeah, sure. As the questions start to come in, let me just go through this takeaway. So what we want to do is establish a preventive maintenance schedule, start performing routine decontaminations, facilitate training and teaching to your personnel, determine the appropriate strategies to minimize or manage the risk of contamination, and then communicate all of those maintenance schedules, decontamination schedules, teachings and trainings, communicate that to everybody involved. And with those takeaways, I think you'll be able to have some improvements, or at least a good preventative maintenance plan for biological materials.
Thanks, Ryan. Some important points there to take away, and the questions are great that are coming in so far. I do want to remind everyone, I saw some people signed in late, that we will have this slide deck and the recording available to you tomorrow morning. So please keep an eye out for that email. We'll also share a link to a short survey. We'd love to hear your feedback on the webinar, any ideas you have for future topics, and improving these. We're a couple a month and plan to continue that. As a thank you for attending, we'd like to offer you a complimentary consultation to help you develop your own biological decontamination plan, where someone from Ryan's team can come out and take a look at what you have going on and speak with an expert. So to learn more about that, I'm sending over a link through the chat box. Feel free to take a look at that as we're going through our Q&A. And finally, on the very last slide is Ryan's email address. Feel free to reach out to him directly if any questions crop up after today's webinar. I'm sure he'd be happy to address those.
Absolutely. Thank you, James.
And now for those questions. Ryan, to start, for preventative measures, how often should a BSC be professionally decontaminated? And that's from Adrian.
Thanks, Adrian. Great question. That's all depends on the frequency of use. I'd say if you're using it daily, I would recommend a yearly professional decontamination with regards to routine operations. But keep in mind that if you're changing the operations completely, and if you're changing your cell cultures completely, you're going to want to probably decontaminate at that point as well. And also keep in mind that the National Sanitation Foundation has some recommendations that it be cleaned if you're going to move it to another room, or if you're going to move it to another facility, or if you're doing any sort of other maintenance on it, like HEPA filter changing. But as far as a routine, if your operations are pretty consistent, I would say a yearly cleaning is definitely beneficial.
Thanks, Ryan, and thanks for the question, Adrian. The next one's from Jason. He asks, what about ethylene oxide disinfection?
Ethylene oxide disinfection isn't something that I covered today, but it is an effective means in certain scenarios. It's quite hazardous, so it's not too common. And I don't have any systems that utilize it as efficiently as chlorine dioxide, but always have to depend on exactly what piece of equipment is compatible and what sort of material you're going after. But with all those things in mind, ethylene oxide can be an effective disinfectant.
Jennifer wants to know, Ryan, what's your opinion on chlorine dioxide tabs?
Chlorine dioxide tabs. I'm not too sure the question. If you want to email me a little detail, we can connect and take this offline. With chlorine dioxide, I use a generation chemical to generate chlorine dioxide gas, which is in powder form, and then utilize that to disinfect enclosures that have been fully sealed. But as far as tabs, I'm not too sure the question. If you want to email me, that'd be great.
Yep, or Jennifer, if you want to send through some more information to clarify, we could take a look. Let's shift to another question. Ryan, what tips do you have for small labs that might not have a lot of resources? This is from Lorena.
Sure. For small labs with not many resources, it doesn't take much really to have a plan with some small, I want to say cheap, but inexpensive chemicals that can be used to clean, like 10% bleach and 70% ethanol, and using those routinely on things where your materials are coming in contact with, but taking it to the next level. Like I said, if you have some equipment that, like an incubator, that can be taken apart, it definitely is beneficial to do that. Have a couple people that might have to do some homework onto how these pieces of equipment work and how they can be cleaned, and assign that person with those tools to be responsible for maintaining them. I think you don't need a full mini chlorine dioxide system to be successful when you have a successful preventative maintenance plan and personnel trained in knowing how to keep things clean up until something drastic happens. So I think with a small lab you can still be successful.
Lola asks, how long should bleach be on the surface to decontaminate the surface? Is it about 10 minutes?
Yeah, it's about 10 minutes. That's the recommended contact time for any of the sodium hypochlorite solutions.
All right, thanks, Ryan. And Josh, following up on that, can you provide any insight onto the shelf life of the 10% bleach, 70% ethanol solution?
That's a great question as well. I know people who use different ones, and that all depends on operations and how routinely that's being used. But I would say no longer than 3 months, and that's if you're using it frequently, you shouldn't extend that shelf life. But if you're not using it frequently enough, then you might consider changing, especially if you have like small volumes, 500 mils sitting on the counter. It's just being exposed to a lot in that time frame, and can start to degrade after that.
All right, we'll do a last call for questions here. I've got one more, and we do have some time, so if there are other pressing questions, get those in. Ryan, at the top, we spent some time talking about mycoplasma. What other contaminants, specific contaminants, should we be concerned about?
Honestly, the best thing I can say for that is just bringing in outside dust and debris and mold, because that's where all that is going to lead to. You know, there's a lot of systems in place in clean rooms just to prevent things from coming in from the outside. Because mold spores are ubiquitous. You bring them in from the outside constantly. That's some of the major headaches just come from environmental mold that is brought in through shoes, or, you know, not wearing a lab coat well, not cleaning it often, regular clothes inside clean areas. That's should be one of the top priorities, I would think.
Great, thanks Ryan for your insight. It looks like we're all set on questions, which is great. I hope Ryan answered all of your questions today. I want to thank you all for attending and for getting your questions in. We appreciate your time. Ryan, thanks for presenting. Do you have any final thoughts?
No, I just want to thank everybody for attending. I definitely I'm interested in this topic, enthusiastic about it. So if you have any questions or you want to connect, my email address is there. Please feel free. I was looking to improve on this topic. So thanks for coming.
All right, great. And don't forget to check your inbox tomorrow morning for the resources we talked about earlier. Thanks, everyone. Have a great rest of the day, and we hope to see you on a future webinar. Take care.